We're going to go ahead and get started. If everyone could take their seat.
So just don't run away.
Great, thank you all. We're going to begin our morning panel, and I'd like to introduce Dr. Katie Donahue. She's the clinical team lead for the Division of Gastroenterology and Inborn Errors of Metabolism at the FDA.
Good morning and welcome. I am so excited to be here. And I have, as we get started, I have no conflicts to disclose, but all opinions are my own. They're not official FDA policy. And I might need help. So part of why we're here is that I sit across the table every day from pharmaceutical companies that are trying to develop a new therapy for patients. And often we are designing the very first clinical trial that's ever been done for patients with this disease. And it's a little bit like trying to build an airplane while you're flying it. It's a challenge. And so we're here today to try and make sure that that endeavor is more successful. Often we're sitting there designing the trial without any real idea of how long it takes for patients to develop some of the more challenging symptoms that affect them. We don't have any of this longitudinal natural history data. That's what you've been hearing all morning. And that time piece is the foundation of good science. Understanding how long it takes for patients to have a change in their circumstances is critical to make a good clinical trial. And some of you who've had the misfortune of hearing me talk before have seen this slide before, but this is what it's like. We have to build our clinical trial before we have our foundation. And so the vision for the Cures Accelerator is to build the foundation. So I'm just curious who is here in the audience today. Can you raise your hand if you are with a patient group? Okay, and what about industry folks? And what about regulator folks? Okay, and do I have any policy and legislative folks? Okay, so any civil engineers? Has anyone here ever built a bridge? Really? Where's my bridge builder? Okay, so we've got one. But raise your hand if you've ever driven across a bridge. So that's what we want to do, right? This Cures Accelerator is scientific infrastructure. We want to build a bridge so that we can help patients sort of be in the driver's seat and take their natural history data and use that to build a bridge that's going to help us launch clinical trials that actually work. And so this is, the vision for this is the pre-competitive space, right? Like we want to do something for patients with the greatest unmet medical need. So these are the patients who have no approved treatments, and they may not even have any treatments that are in ready for clinical trials. So before that's even happening, we can use science to help make things better. So science, I think why I like being a scientist is it takes our suffering and transforms it into hope. One patient's story of struggle is a story. 10 patients' story of struggle where we calculate data and time systematically, that's science, and we can use that to make treatments that work. That's why we're here. So, it's bad when your Cures Accelerator slides won't go forward. All right, there we go. So the first key issue is patient privacy and data security. So at the heart of this, we need to get this right. And my hope is that we would be able to work together and come up with a consent form and data use agreements that are common to the platform so that we're not having to renegotiate this for every single disease, and that we'd have a common language and a common understanding about the kinds of data that are going to be collected and how and when and under what circumstances those would be shared, so that patients have confidence that their data, their story is going to be used to help them. Okay, so who do we need at the table? Patients. We also need epidemiologists. There are never enough epidemiologists at the table when we are talking about designing some of these observational studies, right? So epidemiologists are the scientific experts in real-world evidence. They're the scientific experts in designing these, in conducting these kinds of studies, and in analyzing the results. And they can help us do a much better job of filling in these gaps. We need the geneticists and the rare disease subject matter experts. They know more than any of us about these diseases, except maybe some of the patients. And we need our drug developers. There's information that drug developers need that, and they have a real sense of how they would have to use this data in order to plan a clinical trial. And so we need all of these groups in the room to help decide what data to collect. Okay, so the first thing is symptoms at diagnosis, right? So one of the big changes that's happening that's really exciting is that as our molecular diagnostics are getting better, we're diagnosing patients earlier in the course of the disease, some of them before they're even symptomatic if they're the younger sibling of an index case. Well, this has absolutely massive scientific implications if we want to be able to use real-world evidence and observational data to understand our patients. So we need to know that. How sick were you? What was going on when you got diagnosed? We need the genetic diagnosis. Sometimes you can diagnose rare diseases with an enzyme test or something else, but to really understand what our data are telling us, we need that genetic piece. We need to know predictors of clinical outcomes. So for most of these diseases, there's at least one or two things that we know as doctors, hey, if patients have this, they may have a slightly better time. They may do a little bit better. Maybe it's a certain genotype, or maybe we know that they're working really hard to adhere to their diet and that's going to help them do a little bit better. So for almost all of these diseases, there are a few predictors of better outcomes, and we need to measure that systematically. Background therapy, even though there may not be a specific targeted therapy, all of these patients are receiving treatments, whether it's diet or mobility assist devices, surgical procedures, respiratory support. These kinds of things can all influence patient outcomes, and so we need to really systematically document what kind of supportive therapy are patients getting. Time to event. You've heard this over and over and over again. Understanding the timing before clinical events happen is crucial to planning a good trial, and it's one of the big things that's missing in the case reports that we see in the literature. Endpoints, right? We need to know what would we want to try and use for an endpoint in a trial, and is there a way to measure that once a year in patients in a natural history study? Maybe it's a 6-minute walk test, maybe it's lung function, but there are some commonly used endpoints where if we could get longitudinal data once a year on patients, that would radically change our ability to plan a successful trial. And there's a role for mobile data too. You know, if we're collecting patient-reported outcome measures, well, why can't patients put that in on a mobile phone? And then adverse events. So again, we want to get a sense of what kinds of challenges are patients experiencing just as part of their disease. And that can help us tell a difference between, you know, a side effect that's coming from a drug versus something that's happening just because of the disease. Okay, I put these guidelines up at every talk I get. This is like the good housekeeping seal of approval for observational data. They're called the Strobe guidelines. There's a link right there, and we need high quality data. Most of our natural history data in rare diseases is not adhering to every aspect of these standards. And patients with rare diseases are so generous with their time and in participating in trials, and to me it's just wrong not to do high quality observational studies in these patients. We really need to be collecting gold standard data, and this is how we can do it. And then data standards, right? Just like I don't know how to build a bridge in order to ride across one, we don't want our patients and our investigators to have to know about CDISC and MEDRA and all of these complex data standards. But this is part of the infrastructure you need in order to analyze a clinical trial. And so if we build that structure into the platform in the background, then when it comes time to plan a clinical trial and maybe even use some of this data to support drug approval, we're going to have it in the format we need. And we won't be back coding it and that sort of thing. And we won't need patients to become database experts because the platform will already be built for them. So that's the idea. And with that, I would like to introduce my panelists, and we're going to have a conversation about, these are all drug developers on the front line of making therapies for patients with rare diseases, and we want to hear from them about what are the kinds of information that this platform could collect that we could actually use to speed up clinical trial development. Do you want to bring them on up now, and then I'll introduce them?
Sure.
Okay. Are we all coming up, or one at a time? Alrighty. This is Thomas Blattler. He's had the misfortune of working with me on a few drug development programs. And he is a neurologist by training and chief medical officer at Orphazyme. And all of the panelists we have shown a great deal of courage and creativity in developing new therapies. And next, we have Rose Angel Cruz, who is possibly the most successful of all of us. She is the Director of Research and Clinical Affairs at Cures SMA. And she has had more success than most of us in this endeavor. Next up is Tanya Fisher from Sanofi. She is a neurologist by training, and she's been developing treatments for multiple sclerosis and Gaucher disease and Parkinson's disease. Next up, we have Josh Lehrer. He's the Senior Vice President at GBT. He's a cardiologist by training, and he has really pioneered some of the treatments for sickle cell disease. And then we also have Jeff Sherman, who's the Chief Medical Officer at Horizon, and he has had a lot of experience with rare disease drug development. And so I'm glad to have our panelists here, and I look forward to hearing what you think about how we can use the Cures Accelerator to move treatments into trials and to patients as fast as possible.
Yeah, it is.
So I warned Rose Angel that I was going to put her on the spot and start the conversation with patient data privacy and concerns. And then I do want to hear from some of my other panelists about any of your experiences with that. Maybe it's challenges with having to go back and reconsent patients in order to use data, natural history data, for regulatory purposes. So what are some of the challenges that you've had with that? And I also want to hear from the patient perspective, what do we need to do in order to make sure that patients are confident that their data is going to be protected and used to help them?
Well, thank you, Katie. And again, it's great to be here this morning. It's a little bit of a challenging question, simply because I can speak about my experiences with patients and consenting and some of the things that concern them, sort of given my personal experience and journey working with patients as a clinical trials manager and coordinator, and some of the questions that do come up on that level, which is very much reflective of what we see at a larger organizational level. So I think that a key thing to consenting patients and the kind of information that we need to give them is essentially you have to start with a relationship of trust. So I think that whether it's a PI, a doctor, Cures SMA as an organization and how patients view us, what they do and what we tell them about the data and how we will use it, as long as we're clear, as long as we can explain to them from the get-go why this is going to be beneficial to them as a patient, but also largely to the community, and the benefits of collecting that data in the first place, and reassure them that we will not abuse the use of that data, that is going to be used appropriately as stated on the consent. I never really did have a problem with anyone saying, yes, no, I don't want to consent. And I think information is important. Clarity is important. Use of data, why is it relevant? So I think those are all important things. I think it's a challenge for us. We are working on a clinical trial registry right now, where we're working with key hospitals in the United States who are experts at the care of SMA, and we are collecting a plethora of data having to do with both preclinical and postclinical data, post-marketing data as they call it. And so some of that will involve reconsenting the patients because we're collecting a set of data that it's for a new purpose than what was originally collected. So, we're working the kinks. I'm not personally involved in that project, which is why I was hesitant to talk about this. Dr. Schroth is our Chief Medical Officer and is very much involved in the collecting data for patients for purposes such as this. So I don't know if that helped.
What about my other panelists? Have you had any experience with having to go back and reconsent, or challenges with consent issues, or patients who thought that they agreed to one thing and then data was used another way? I mean, we've certainly seen this at the FDA without specifics, but this is a real challenge. Yeah.
So have had the opportunity working through the FDA Clinical Trial Transformation Initiative, looking at best practices for sponsor-patient interactions. Part of it is to really proactively try to preempt those issues by really getting the patient groups involved as early as possible in the development pathway, to really look at what are the important endpoints, really think about the end in mind, which is certainly getting a product approved and available for patients in need, and really going through what is the data that needs to be collected, what can we do in terms of informed consent, et cetera, to make sure we get the data, but that their identity and other things are really protected, so everyone's really part of the whole process and journey, rather than to go back and clean things up, so to speak. And then, it's all part of this initiative as well. What can we do to really leverage those learnings and really make sure that's available for everyone? So hopefully, live in the Chicago area, we can avoid potholes, so to speak.
One example, just following up on that kind of specific example that I've seen is around there's a bit of a tension, because we talked about sort of how much is sometimes unknown in these rare diseases, and you want to cast a broad net, and even sometimes registrational trials have a lot of exploratory intent in some of the assessments, and then that can create a challenge in explaining to a patient what is the actual purpose and why is this valuable and why is it worth them, either, especially for example, DNA samples. So we had sort of some specific experiences where I think the learning was kind of going back and being able, whether it's a consent form or making sure that the PIs can explain in a very practical way, what is the impact of this? Understanding, for example, how your genotype predicts your response might actually help us treat you better or figure out the dose for you, rather than just have something in a schedule of assessments that says, something that looks scary, like DNA sample. And then patients are kind of worried about what does that mean. It could be in a freezer for years, and who's going to test it and for what. So I think being, not compromising the scientific exploration that has to happen to develop these therapies, but also being able to explain with enough specificity to patients and participants why, why these are thoughtful and why these are important assessments.
Yeah, and the other thing to add on to that is that I think we focus a lot on the U.S., right, because that's where we are right now, but different countries have different regulations. So, for example, if you want to bank DNA for exploratory purposes down the line, and I can tell you I've personally had this issue in one of my trials, there are some countries that they'll let you do everything else but bank the DNA for that reason. And so you lose a cohort of patients that may be very valuable, that can add to the overall richness of the data that you are collecting. And so you just have to be a little bit mindful of what you can and cannot do, and then think through what is the overall effect of not including those patients and how it impacts the overall data set.
Very recent experiences that we've made, we're trying to help a patient registry rescuing data from a sponsored registry. And so one of the big challenges is reconsenting patients who died already a decade ago, or reaching out to parents to do that. So it again, to me, speaks very much towards having a disease registry that is not owned by a sponsor. And the moment the sponsor decides to pull out of a disease area, you just lose valuable data. So I think this very much again speaks to trying to get those data into the pre-competitive space. These data are extremely valuable, and it's just a shame when we lose those.
Okay. And then what are the biggest knowledge gaps that you see? So when you've got a promising candidate therapy and you're ready to come in and talk to the FDA about starting a clinical trial, what do you find are the biggest unknowns that make it hard to do that? And what are the gaps that we need this platform to fill?
There's over 7,000 rare diseases, only 5% of treatments, and part of it is really what we're talking about here, really, and everything that you went over, Katie, in your remarks, Teresa, et cetera, which is really knowledge of the disease state, natural history of the disease, what are really the best endpoints, how best to collect the data. You almost have to think about it with the end in mind. If the end in mind is really regulatory approval, what's really going to be needed to really facilitate that approval? You know, and in some disease areas, there may be more knowledge than in others, but what can we do to have one kind of a level platform that really reduces barriers for companies to really do development in some of these very needy, you know, diseases that are life-shortening?
I think, again, just to build upon that, I think that the struggle, at least the struggle I have, is that in certain diseases, there's a fairly rapid progression of the disease. So you can do, and if they're in children, it's fairly easy to figure that out, right? It's developmental milestones, it's time to event, time to death. Like, those are real hard clinical endpoints that you can do in a fairly short period of time. However, if you do certain rare diseases that are in the more, you know, juvenile or adolescent or even adult state, those endpoints can be quite long. That progression can take years. So then you have this fine balance between doing a traditional clinical trial design where you're asking a patient to potentially be on placebo for 2, 3, 4 years, which a lot of patients won't do that, versus trying to do something where it's appealing for the patient, it's doable from the sponsor, and it gives enough information that the regulators are comfortable saying, yes, this is clinically meaningful, or yes, this has made a difference and we approve, versus no. And I think it's a very hard balance. I don't think anybody, whether it's patient groups, patients themselves, or the industry sponsors, you know, we all want to do good, right? We all want to make this work, but it is very hard to be able to generate that data in order to be able to make these very hard conclusions. And I think this is, again, understanding the natural history progression. And again, I'm being selfish here, not just in a very fast progressing rare disease, but in some of these diseases where it can take years to show a real big change in progression is tough.
Well, I mean, I think what Tanya raises is the whole, are there markers of diseases, biomarkers, who's developing those, have they been validated, not validated, what can be done, and then the sense of urgency to really move development forward because of patients in need who are succumbing to these terrible diseases.
Yeah.
And I think along with the understanding of natural history and the need for longitudinal data, I think some gaps that sort of we're always thinking about is the rate of change of patients over time. So like you're saying, you know, and we don't, right now for SMA, we have 2 therapies that have been approved. We're very, very thankful for that, but we don't really have good endpoints for adults.
Yep.
They are being dosed, they're benefiting from treatment per their clinical meaningfulness responses and the way they're interacting and reacting to the treatment. However, it's hard, the endpoints that we are using currently are not sensitive enough to pick up the subtle changes that to our patient population is meaningful. However, when you're thinking about a clinical trial, like you said, timing is of essence. You don't want to put a patient, especially if it's an infant whose, you know, life is really at risk within, you know, 10 months of life on an extended placebo-controlled trial, because it's not ethical, and parents would likely not go for it. So you want to understand at what point within the disease process are patients changing most rapidly? At what point are they more likely to respond to certain interventions? You know, at what point are they stable versus going down? So all of those things are important in terms of designing your clinical trials timeline and the expectations for treatment, as well as the endpoints that you use to measure those changes.
Well, and this is where collaboration with, you know, leading institutions is really helpful. You know, at Horizon, for instance, we're collaborating with Children's National, and they're putting together the first Rare Diseases Institute to really look at the whole spectrum of drug development from start to finish and what can really get done. And they have expertise that a company or a smaller company may or may not have. And then really looking at a number of the umbrella patient groups and other things, whether it's NORD, what Critical Path is doing, Global Genes, Rare Disease Forum, other groups as well, on what we can do collectively to move this whole area forward. And I think you could even talk about what FDA has done in terms of some of their grant programs for rare diseases to help move this whole area forward as well.
I think here it's really also important to emphasize the pre-competitive space where companies can actually work together. We have a
Absolutely.
mutual interest here, be it to actually better understand how disease progresses, better understand how we actually can measure progression, and third, actually also understand what are the factors that predict different paths of disease progression. And I think this, in particular, when you're in very small, very rare diseases, the heterogeneity is probably the greatest obstacle to success when developing drugs. We need to understand how are the different trajectories. We need to understand, if we do a placebo-controlled study, we'd ideally have a placebo group that really compares to the active treatment group. But if you have like 16 and 30 patients in each arm, well, you guess what? What's the likelihood that those two groups will not be very comparable? And then it'd be very interesting and very great actually to have the knowledge of the disease. What are the factors that contribute to differential disease progression? And can we use those factors, for example, in a covariate analysis where you say, okay, once we have the data, can we actually correct for those differences?
Exactly.
In particular, when you have those small groups. And that's where an initiative like this is invaluable.
So I'd like to hear more from the, go ahead.
Yeah, I mean, just thinking also what Dr. Woodcock mentioned, which is that I think everyone in this room wants to speed the development of new therapies for so many of these patients who don't really have good options, and that one of the biggest challenges is really tremendous variability in disease progression in patient populations, and that's compounded in rare diseases where you have small populations. That variability is sort of an additional challenge, and endpoints were talked about, and without, I mean, can't do anything without good endpoints. You know, in the experience that we've had working with FDA in sickle cell specifically, which is a larger rare disease, so we do have access to more patients. But I mean, as a cardiologist, I came to this field and was kind of surprised that the endpoints looked a lot like a cardiovascular disease, where we have 20,000 patient studies and can overcome the variability in pretty subjective clinical endpoints. And we did a lot of one-on-one work with the division, but I think can be leveraged in this context with sponsors working together to go back and look at legacy academic databases, to build the support around a subpart H argument that there could be endpoints that would really address some of that variability. It would be predictive of long-term response, but could be modified much more rapidly in a trial. And leverage, I think, what is common across many rare diseases, which is one of the advantages that we have is often we understand the biology better. We understand the mechanism better than in larger primary care type indications where there isn't a genetic basis clearly defined. And so how do we translate that? How do we take that knowledge and use it to make better endpoints which we modify more rapidly? And then we show that predicts long-term response. And the only way to do that is by pooling together resources and having better long-term natural history data where you can tie early changes in those biomarkers to longer-term outcomes.
Right. Yeah, I want to invite the rest of the panelists to comment on that because I think that's one of the biggest gaps that I see. So, as Josh pointed out, one of the few advantages we have in rare diseases is that often we have a very specific understanding of the biology, and often there is a biomarker that's very specific to the disease, but it's primarily been used for diagnosis. Does the patient have the disease or not? What we almost never have is high quality data showing us that this much change in the biomarker is going to predict this much improvement for the patients. That is a huge missing piece and a big obstacle to agreeing that a change in the biomarker is going to result in a clinically meaningful change for the patient. And often the question is how much. We usually have no idea how much change in that biomarker is it going to take to make a difference for patients. And so that's one of the knowledge gaps that I'm hoping for that the platform could address. But I'd really like to hear specifically from the panelists more on that topic.
From my perspective, yes. I mean, particularly when you develop drugs that should be modifying disease course, you want to start as early as possible in the disease course. And that's typically when the patients don't progress the most. So the conundrum is, well, now we want to start early, but we can only show an effect 5, 6, 7 years later. And that's what you're absolutely where we face the challenge, where it would be great at least to, first, do we have a drug that has an effect on that biomarker? But then we actually really need 5, 6, 7 years of follow-up to actually really understand how that biomarker predicted treatment success. And this is one, to me, is one of the greatest challenges that we face nowadays. Also, where trials need to be shorter and shorter to actually also engage patients in participating in a trial, and the actual clinical effect is seen way down way later than that observation period. So here again, registries would be tremendously helpful to see how an effect early on may predict outcome later.
Yeah.
So one challenge is some biomarkers may be predictive of disease progression, but are they modifiable by drug treatment?
Yeah,
And so that's a quandary. And then are those biomarkers readily available if something's a CNS disease? You know, how able are you really to measure that substance? And you know, this is something I think that FARA's been looking at in Friedreich's ataxia, where you're dealing with a central nervous disease, but then the marker frataxin may or may not be readily available, and how predictive is frataxin in other tissues.
But even if to that point, even if it's actually, let's say neurofilaments are a big deal in SMA, and we're looking at, intervention actually makes the levels of neurofilaments decrease quite dramatically pretty soon after the patient is treated. However, what's not fully understood is, what does that mean? What does that, how does that predict the course of the disease and how well the patient is likely to do over time under this treatment? So it's just not that straightforward, and it really takes a lot of observation. And a lot of times we have really good animal models that are good in identifying, say, a biomarker and giving us a hint into how that might work into the biology of humans, but it's not necessarily translatable on a one-on-one scale. So, and it's also resources. I mean, biomarkers and testing patients over time and analyzing samples, it takes a great deal of funding to do that. And so that's also a challenge for rare disease, and where people may or may not be, people meaning pharmaceutical companies, may earlier on not be as interested or invested until they really know there's something that's more promising down the line. I think that's the challenge of.
I also think that, at least in my mind, I'm an N of 1 here. We have to be a little bit careful when we say biomarkers, because there's biomarkers and then there's target engagement, right? And then you can use something like target engagement to help supplement your clinical endpoints. And that can help with a whole host of things. You can show that if you really understand the mechanism of action of your drug, and you're really sure that it's able to engage the target that it should be, then you have a little bit, I think, more to work with, at least in my opinion, because then if you're able to show that there's some target engagement, then you can kind of piece together, well, what would I expect the clinical endpoints to be? And I think if you can make a scientific, rational explanation of that, then that may help kind of move things down the road. But not all diseases have that ability, and not all drugs have that clear ability to show what is it supposed to do and where is it supposed to work. Right? I mean, some of it is, it's just not, it's not there. And I do agree that unless, at least from a company standpoint, everybody is super excited, and everybody truly believes in the programs that we work in, and they want to make it work. But until you really have that proof of concept, you're not going to pour in millions of dollars into validating a biomarker, because it's a lot of work to validate a biomarker. And it probably, again, in my N of one opinion, it probably shouldn't be one company that's doing that. Right? It should be a collaboration that's doing it, because no one person should really own this.
Then it's really making sure there's resources available to help facilitate that, because a lot of the companies in the rare disease area may be very small biotechs that really don't have the resources and are just trying to pull as much funding together to even get to an IND stage,
Yep.
or even get to a readout of proof of concept, and that's part of the challenge. But that's really where, again, whatever we can do to pool resources to help one another, because then at the end of the day, we all benefit, because that patient could be any one of us.
Yeah, exactly.
I completely agree about the comment about sort of the challenges and opportunities of biomarkers in some of these diseases, but I think it is an opportunity to leverage one of the advantages, which is understanding the biology and mechanism, and that not always, but often even markers of target engagement have biological relevance.
Exactly.
And then it may not be that heavy a lift if you have enough longitudinal data to to build the evidence around that biomarker, not necessarily to make it a fully qualified or fully validated endpoint, but the standards are different in the case of large unmet need and the Subpart H pathway, for example, and accelerated approval, but difficult for a single sponsor to kind of collect that kind of data.
I mean, some of these diseases, like we talk about doing like a 30, 40, 50 patient trial, and people, like from cardiovascular, you're like, wow, that's amazing, right? But to find 30, 40, or 50 patients can take a long time. And you have to go to a lot of countries. And it may not be enrollable depending on inclusion and exclusion criteria and length of time that they're going to have to be on placebo. And so you think that, I can do 30, 40 patients, 6 months, year, no problem. And it's 2, 3, 4 years. And that's hard. It's hard on the company. It's hard on the patients. It's hard on the sites and the PIs and the whole team that's at the site. It's just tough.
I think something that's interesting, it just came to my mind, is exactly when you have to follow patients for a long time, you will have patients who get lost to follow.
Yeah.
And now that's again where I think a registry becomes important, where you actually can compare who did I lose there. Is that a patient I just lost? Of course, you always try to figure out why they got lost, but you always end up, you start with 50 patients. After 5 years, you have, if you're lucky, you have 10 patients, right? And then you say, okay, how representative are those 10 patients of the 50 that started in the beginning? And I think that's again where we actually also can go back to registries and try to compare and figure out.
Yeah. And this ties into my plea to have epidemiologists at the table, because this is what they do for a living. So when they're building a registry, they think about the potential for loss to follow-up, and they think about how could that affect what we're able to do with the science. And there are deliberate strategies for maintaining and supporting patients in their participation. And I think we have a lot to learn from them in the rare disease clinical trial space about how to do that. And that's part of what we're going to need to do in order for this data to be useful, is if we get patients to contribute one round of data, that's great, but what you're hearing from us is that it's the data over time that's really going to accelerate cures. And so we're going to need to put patients in the middle and make sure that the data we're asking for is the important stuff, and that we're not asking for so much data so often that it's a real burden for patients. We want as many patients as possible to participate, and that's going to mean really keeping an eye on feasibility and burden for patients in terms of involvement.
Well, that again reinforces why having the patients involved from day one to really address the issues of what may be meaningful, what's not, what are the barriers to really be able to participate not only on day one, long term as well. And it's also getting the family members, the healthcare professionals who may be carrying them, a whole group together, because it's not just the burden on the patient per se.
So I just want to add, I have a really pretty extensive experience collecting natural history data through sort of my work at Columbia University. The Pediatric Neuromuscular Clinical Research Network, or PNCR, essentially began collecting around 2008 or 9 the first set of natural history data for SMA patients, well before there was a therapeutic really readily available to be tested in clinical trials. They understood that if we wanted to move forward in that direction, that we needed to collect that information and have the understanding of what we've all talked about, the progression, natural history, the various phenotypes, the different outcomes through which we would evaluate the progression of disease and how well the patient was doing or not, et cetera. So what made that very possible and sort of was essentially they combined a clinic visit and sort of paralleled the visit with the same things they wanted to collect for the PNCR data. And so in that sense, there had a couple of exploratory stuff like, the c-map, a few other markers. But eventually, a lot of this information that we collected was just part of a, regular medical exam. So it wasn't too burdensome for the patient. Everybody was getting consented and collected for that. It was just part of what I did for clinic. And so, we had patients that they'd come to clinic. They have to because they have the chronic disease. So I think if you're well organized and you involve healthcare professionals, if you involve the patient community, and everybody's sort of on the same page in terms of what's needed, that's probably the best way for you guys to sort of collaborate and collect that data and funnel it into one place. But it shouldn't be like, oh, we have to do this clinical trial together, otherwise it's just really hard to do.
I'm just going to mention that there are a lot of reasons why doing that makes sense in a pre-competitive space, as was discussed, and resources were discussed. But I'd be curious to get other panelists' take. I mean, the resource issue is a big one in that in our experience in sickle cell, we were at the patient-focused drug development meeting 5 years ago now, and came out of that deciding, well, there's a real opportunity and really kind of an obligation to think about new endpoints to get to the unmet need and to get around some of this variability. But in doing that, trying to develop a new clinical outcomes assessment, as a single sponsor, trying to qualify new objective endpoints that would be tied to clinical outcomes and be suitable for potential registration. I mean, those were almost like having entirely new development programs. I mean, in terms of the resources involved, and especially for a small biotech company, there has to be such incredible amount of focus. And it's not just whether you can even raise the money. It's focus on moving that drug forward, and to have these kind of obligations going in parallel as a single sponsor working one-on-one with the division it's a really tough climb, and it creates more risk and not just cost, but I think more risk that will actually end up ever getting to patients.
Yeah, what we found to overcome some of those hurdles is really work together with the patient group, and they provide sort of the nidus to bring some of the companies together, researchers together, to really look at what can get done collectively to move the field forward.
I think Cure SMA has done a fabulous job in the past years to actually pull those different players into one to your research roundtable, and actually to make sure that those sponsors meet in a pre-competitive space and work together, because I think that is one of the biggest challenges. I figure it must be also frustrating for FDA. You have 2 or 3 sponsors developing their drugs in a given indication, and everyone comes with a different outcome measure. So that's where I really see the need also to actually work together here and use the registry as a vehicle.
Yeah, the incentives are not lined up well when it comes to patient-reported outcome measures under our current regime. So I'm really glad to see that these are being included as part of the Cures Accelerator, because it is a ton of work to develop and validate a patient-reported outcome measure. And I've seen this not so much in the rare disease space, but in some of my other work, where we've got multiple sponsors developing multiple different patient-reported outcome measures, and they're making them all proprietary, which means they've done all this work. And from a business standpoint, I understand that they need to do that, because if you make it publicly available, it makes it very easier for your competitors to come in. And so from a business standpoint, they can't do that. But from a drug development and a patient standpoint, this is terrible. You know, we want one really good patient-reported outcome measure that gets used consistently and that we can see over time and across multiple programs, and we have confidence that we know how it's working. That's the kind of patient-reported outcome measure that's going to give us the best information to patients and doctors about, hey, how is this drug going to help? Do you want to take it? And having really consistent measures over time. And so I'm thrilled to see that piece included here, because it shouldn't be on any one sponsor to do all of that work. And then it shouldn't be one sponsor that then owns this patient-reported outcome measure, because that's a lot of patient effort that goes into it to contribute and to help develop the measure. And that information is too valuable to be sort of siloed in one sponsor, I think.
Yes.
And for patient-reported outcome measures specifically, I mean, there's a lot of reasons why I think a lot of stakeholders are interested in integrating that into clinical development more, and it makes a lot of sense. But I think even if an individual sponsor does go through that process and qualify, a PRO, something that is measuring symptoms that matter to patients, the likelihood of being able to kind of sufficiently de-risk that to be kind of practical from a drug development standpoint is fairly low. In that you can, you go through the process following, with FDA and qualify, but is that instrument actually sensitive to drug effect? Is it modifiable by a treatment? And one way to get at that is actually, to combine the development of PROs kind of across sponsors with collecting data on those instruments in registries or with long-term natural history, where you can at least kind of triangulate. You could say, okay, well, patients with this genotype or this subpopulation, where we think our drug affects that same biology, that reads out in the PRO, or some other standard of care therapy may show changes on that same PRO, and then it would make sense to use that as an endpoint in the trial. But short of that data, it's a very big risk, even if you can go through the whole process, to actually put that into a study, having no idea whether you're measuring variability in the disease that a drug can affect or not.
Well, some of it is even the timing on when those are really developed. Because usually what happens is companies get involved in a clinical trial, and it would really be great to measure something patient-reported outcome. And is that just a bolt-on versus we've really thought about that, really worked through all of the questions, validation issues, et cetera, and then they wonder why they're coming up short when it comes to a review. So it's really, again, what can we do to help enlighten everyone about the earlier we start in the process, hopefully the better the outcome we'll have.
But I think also, just a thought popped into my head. It's not only thinking about it from a company or sponsor standpoint, but it's also helping to level set things for patients and the patient groups. Right? Because sometimes, I don't know if it's been anybody else's experience, but sometimes the patients groups come and say, Fisher, what are you doing? Like, why aren't you, measuring how we're feeling or how we're getting better or how we're doing something? And you need to kind of level set, well, we don't have the tool right now, or we have to develop the tool, and then when you kind of explain things, the patients say, oh, well, we can mobilize, we can try to do this. And sometimes it just kind of gets to get that conversation going is sometimes what you need. And then once people understand what's needed, then, movement can happen. Or sometimes it can't. And that's okay too. At least you begin that conversation and you understand what can and cannot be done.
Well, I mean, what you raise is that the patients are probably the best resource and the most underutilized resource.
Yeah,
it's
exactly.
usually oh wow, we'll reach out to the patient group because we're behind in enrollment in a study that,
Exactly.
they really didn't look at and maybe measuring the wrong endpoints, and then we wondering why we have a problem.
Right.
So, in your experience with rare disease research, what have been sort of your biggest surprises scientifically, where you designed a clinical trial one way, and then you found out partway through, oh my goodness, this was a surprise? And how could this platform, this Cures Accelerator, help fill in some of those gaps and make it a little easier to develop a clinical trial?
Well, I can start just briefly to follow up on the PRO discussion. I mean, that was a case where, again, actually starting out fairly early, talking to patients, and sickle cell disease specifically. It's a very symptomatic disease. Pain is kind of a hallmark of the disease, but there is also fatigue, and so that kind of led us to work on developing a PRO in parallel with our drug development program as a clinical outcomes assessment. And also basically learning from patients that the traditional kind of endpoint is really an access to care endpoint. It's basically when a patient is desperate enough to go to the emergency room and often see a treating physician who doesn't know them or their disease and wait and hope that they get pain medication, versus the day-to-day experiences where there's actually a lot of this happening at home. And that was the impetus, and it made a lot of sense. And we thought we had a very rational approach, even though some of the KOLs actually cautioned us that this is more complicated than you think. So, I mean, in that case, we did go through the whole process, which I was kind of alluding to. And I mean, I think the surprise was that we developed an instrument that was kind of very good at measuring the day-to-day episodic nature of sickle cell disease. We'll probably end up publishing a lot of this data. I think it will inform a lot about the patient experience, but turned out not to be a very good endpoint, precisely because it was measuring a lot of the episodic variability, much of which is not modifiable by drug, much of which can be contributed to by so many factors, including in adults, kind of fixed damage, which has occurred over cumulative damage, which has occurred over years. So that was a surprise. I mean, fortunately, we designed sort of a somewhat adaptive trial where we weren't kind of betting everything on that working, and we could end up using a biologic biomarker surrogate endpoint in its place. But that was when we invested a lot of time and energy and collaboration with patients. A lot of patients enrolled in these studies that were really just for the clinical outcomes assessment. And in the end, we learned more about the variability of the disease than about our drug.
Yeah, I would add on to that, studying again Friedreich's ataxia, and I know Ron Bartek's going to be a speaker a little later. It's really looking at a lot of the nuances on how you measure, when you measure, in a lot of the neurologic diseases, if you're looking at walking, etc. Some of those things may be effort dependent. You're asking patients to come in. Do you bring them in the night before so everyone's well rested? Do you start some of the work the next day? Because that could really affect outcome measurement, variability of the measurement, which has an implication then on sample size, etc. So, I mean, yes, the harder the endpoint, a little bit easier in other areas, but if you have something that could be somewhat effort dependent or variable, you have to really think about all the little details and almost really walk through before a study starts, almost like a mock, taking a patient through to really get a better understanding of some of the nuances, because it's sometimes the devil's in the details.
Absolutely. Just quick thought about how you mentioned the devil's in details. Another unintended consequence was that just by using a clinical outcomes assessment in our trial, we had an ePro device with a daily recall. Completely unintentionally, we ended up sort of skewing the types of patients who were enrolled by investigators. So the investigators thought
we're more tech savvy than
and sort of which patients do I have? I have a few hundred patients, but I think only 3 will be able to kind of stick with this, doing it every day for the next 6 months. We actually ended up selecting for patients who had low scores, sort of low symptom scores to begin with,
right.
and made it difficult to actually measure a treatment effect. Yeah.
Yeah, I think also in some of the diseases we go in, there's no endpoints. So I can tell you I oversee the Gaucher type 3 clinical trial. There's no endpoint for us. And these patients are super heterogeneous. And it depends on what country you're in and area, and and there's only a few hundred patients worldwide. And so it makes doing a clinical trial with a traditional endpoint or a traditional trial design super, super, super hard. And so you spend a lot of time in your phase 1 doing you kind of what's the expression? You throw the spaghetti against the wall and see what sticks. I mean, it's kind of what you're doing. You try to see is it this biomarker? Is it this endpoint? Is it this PRO? What do I got? And having a consortium like this to really help, kind of get it all out there and collect it initially, so then whoever wants to use it to say, okay, this, again, I go back to what is my drug and what is the mechanism of action? Okay, this is what it's supposed to do. So what's important to the patients and what is changing in the patients, and does it make sense to use this drug in that patient population? And that's really, I think it's really important and critical.
And this is really the shared learnings.
I'm jumping in. Thomas has been trying to jump in for several minutes now.
Okay, no, thank you. I just want to address your question. You asked one of the biggest surprises.
Yeah.
And to me that was running a trial in 50 patients, ultra-rare disease, autosomal recessive disease. We detected 33 novel mutations that hadn't been described before. So a third of all the potential mutations these 50 patients could have were novel. And I think that's also addressing some of the needs for a registry where you hopefully could more systematically or have just more data on mutations. And it's a very simple example where I see this would be huge.
Yeah, for sure.
This is the importance of really sharing this information, because it's one of those situations where at the end of the day, it's sort of, geez, if I knew then what I know now, what I would have done differently, and unfortunately may not have, the company may be gone by then, but at least to share the learnings so someone else does not have to go through that, the nuances, whether it's sickle cell, whether it's cardiac, whatever that disease is, to really know and have that knowledge could be invaluable.
Yes.
And even just think about, the repository of knowledge that the FDA is sitting on, but because of confidentiality and other issues, may or may not be able to share.
Right. Yeah, that's a big part of the problem. So, Emma, you're telling me we're moving to a Q&A from the audience members? Okay, so what questions do we have from members in the audience?
Oh, don't be shy.
We've answered them all. Yeah.
So you guys talked a lot about genetics and how, you guys talked a lot about genetics and biomarkers and how that could help with diagnosis and treatments. What are you guys' views on genome sequencing as a preemptive measure to find these diseases and the root cause and how to treat them most effectively?
I think one is is that information readily available for a particular disease state? And this gets back to the sharing of information as well. Because there's a lot of tools and modalities out there that sponsors may or may not have access to, or that information, and that's, I think, the whole purpose of making sure we're unlocking, these key, not only data repositories, but tools.
I think it's, you raise an important point. I can tell you my experience with Parkinson's disease patients with a GBA mutation. So Sanofi is the first industry-sponsored clinical trial in the genetic form of Parkinson's disease. So we had to kind of grapple with what you're saying, because if you have a GBA mutation, if you're homozygous, you get Gaucher disease. If you're heterozygous, it increases your risk of developing Parkinson's disease. Doesn't necessarily mean you get it. It increases your risk. So we've had to really start thinking very carefully about what does this mean to the patient. So we give genetic counseling to patients before they get tested to see if they do or do not want to know the gene. All the patients obviously have Parkinson's disease, but it's making us think, if this drug works, what happens down the road? if people have a GBA mutation? What does that mean? What do we tell people? How do they access this information? So we've been working very closely with a lot of the consortiums, PPMI, AMPD, CPP, to really start thinking about this, how as a community, not just as Sanofi, but as a community, what do we tell people? Because there's more and more clinical trials now that are happening in genetic forms of Parkinson's disease, not only GBA, but also LRRK2.
Thanks so much for an exciting panel. I filled 2 pages of my notebook, which is a new accomplishment for me. I work in patient advocacy at Sanofi Genzyme, so I happen to know Dr. Fisher. I think something that was raised was sort of, what do you do when you have really ultra-rare conditions, right? And they're tiny patient populations. And so I'm curious if you possibly see this registry as a way to connect patients to others around the world when they may not know that there's another patient with that condition that exists, as sort of a service to the patients who contribute.
You know, I hadn't thought of that. And I think you'd have to deal with, I mean, would it be like an online matchmaking app? You have to opt in somehow. You don't want to out people who don't want to be outed. But certainly, that's certainly something that we could consider. You think there'd be a lot of interest in that from patients? Yeah.
I think so. I think for one of the rare diseases that I work in, you know, there's a US group and then there's a European group, and they don't really talk very frequently to each other, but I needed that data, right? And so we kind of help facilitate those discussions. And what we've actually seen is that by combining, like by bringing these groups together, you know, we've also been able to bring together, you know, researchers that necessarily kind of knew of each other but didn't really know each other, and that's created new collaborations and you know, work products, and it's nice to see that you're able to kind of facilitate the learnings and moving, you know, the science forward.
So I think one of the keys here will be publicizing this and make this available, because what will happen then, you know, in any disease state, individuals will be searching the internet or whatever, they'll find, you know, this information is available, and then there'll be a number of what we would call unsolicited outreaches to say, geez, how can I get involved? How can we contribute? And hopefully that will even raise this to a higher level.
From our perspective, we'd certainly be very interested in having a more global perspective. I suspect one first needs to show some success here before you expand, but in principle, one of our big challenges is always you have this initiative in Europe, you have that one in US, and now how the heck do you kind of bring those two together and also have comparable data? So we'd be very much interested in having a more global perspective, but I think this is a great start.
Yeah, exactly. All the way over there?
Thank you. Klaus Romero with c-path. I want to come back to the point that Tanya raised, which I think is a very important one. And by the way, you're not an N of one, you're an N of many, and you can count me in. The point you made about the genetic variants in Parkinson's disease, as you know, in the Parkinson's Consortium, we have found some pretty interesting results based on those genetic variants in terms of the disease dynamics, not just about the certainty of somebody getting to a point of getting a clinical diagnosis, but what happens later. And it has been pretty surprising. So can you elaborate a little bit more on the incorporation of those sources of variability to understand disease dynamics and then drug effects?
I know that's a very good point. I mean, I could talk for days about GBA, PD, and lysosomal storage diseases. So GBA PD is actually, if you want, I can tell you the story at another time. I mean, it's from a scientific standpoint, I mean, it's a beautiful story. And it really shows how certain case studies really brought this whole different idea of a new subgroup in Parkinson's disease. But really, you know, to bring it to kind of simplify things, in Gaucher disease, we talk about mild and severe forms of Gaucher disease. Mild are those patients that have Gaucher type 1, the non-neuronopathic form of the disease. So it's peripheral problems, spleen, liver, blood, skeletal, etc. And then types 2 and types 3 are the neuronopathic, or the severe forms. So they have the peripheral manifestations of Gaucher disease, but they also have CNS issues. So why are we talking about Gaucher disease and Parkinson's disease? Because if you fast forward about 12 years, what we found is that if you have a GBA mutation and you're heterozygous for this mutation, it increases your risk for developing Parkinson's disease. And so groups, like you were saying, you have all these different genetic variants. So they were saying, does it matter? Like if you have a N370, if you're a carrier of N370S, which is a mild form of Gaucher, what does that mean for Parkinson's disease? Or if you have an L444P mutation, which causes type 3 Gaucher disease, what does that mean for Parkinson's disease? Well, actually, what it means is that the severity of the mutation seems to correlate with disease progression. So the more severe the mutation is, the faster you progress. Whereas the more mild mutations, the slower you progress. It's still faster than if you were a sporadic patient and had no mutation, but it's slower than the severe mutations. And so it's some of these lessons learned that we're trying to differentiate and to see, well, where does this drug that we're developing, where is it going to hit? Where in the progression? Do we only target those severe mutations? Do we target those with mild? Do we target everybody? And it's this fine balance of what we're doing, of what to do. So it is exactly what you had, the house upside down, so we're trying to learn as we go, which I think is actually kind of fun. But it takes a lot of work, and it's not just us that's able to do it. It's because of consortium and consortium help and pooling together the patients, the patient voices, as well as all the different clinicians who see these types of patients to say, this is something that matters, or this is an outlier, or it's something we should think about, but let's hold off on it later.
It's a great example, and I think really applicable across the board. I mean, we're talking about variability and how do we overcome that, and understanding kind of key genetic modifiers of disease progression, phenotype. I mean, it's very hard to design the right trials and choose the right endpoints without understanding that.
I think modifiers are also important. So you can have two patients with exactly the same mutation that are very different in their progression pattern. So that to me is the next step also towards your question earlier, where we do the whole genome sequencing. So in principle, what we're interested in is, of course, also understanding some genetic modifiers that can have a huge impact in how the patients actually progress.
And who may be more likely to respond to a therapy versus not.
I think one key lesson that I guess just hearing all of us just speak and go back and forth and talk about all these very important aspects of drug development. Like not one is less important or more important than the other. And I think one key lesson that my boss, Jill Durecki, who is the chief scientific officer at CureSMA, told me to share with you guys is that in essence, really getting all of the KOLs, the patient community, everyone involved in this whole entire process is absolutely key. Like now that we know that the voice of the patient is absolutely essential to developing important endpoints, to the way that the regulatory authorities will be analyzing efficacy in a data or deciding whether or not to approve a therapeutic. Let's all learn from those and get together and learn each other's languages. Because I think one thing we do often, especially in rare communities, is we work in silos. We have the patient advocacy organization doing their community survey, and we have the doctors doing their clinical data, and we have the scientists doing their preclinical data. And a lot of times, there isn't a forum for all of them to come together. And I think one of the things that CureSMA has done extremely well is their just holding their annual conference where they get every key stakeholder within the community essentially communicating the progress within the field, having conversations, learning from one another, and essentially learning what's important to the entire community. Because there's not many communities in SMA, it's one community, and we're all a part of that. And I think that's one thing that CureSMA as an organization has done this for 35 years and has 2 successful drug programs that have been approved and are available to a patient community. has done really, really well. So just that I'd share that.
So, what I would say, I think from this panel, it's really a call of action for all of us to really get involved, participate, and share.
Yeah.
If nothing else, because we'll all be better off for it.
I think what's important, this also applies to us as biopharma.
Absolutely.
And that should be our ethical obligation to share our insights that we've gained with valuable patient data with the community.
You have any more questions from the group? Comments? All right.
How do you guys want the community to get involved? Like, it's one thing to say we need the community to get involved, and you guys have this initiative as drug developers, but to get the community and patients involved, how do you expect that?
Well, if I take the first step, and I think you've heard from several of the panelists, we are involved with the community already. So we are talking to patients, to parents, to advocacy groups. We are joining initiatives to try to also to get into this competitive space where we have a huge mutual interest. So I think several of us have very likely also conducted protocol design meetings with parents and affected patients. So we do actually, and that's probably also important for you to know, the biopharma world has moved over the past decade. So we've learned some of our lessons. So we've learned that we need to involve the patient and their carers in developing drugs for those rare indications. So we very much reach out also to, I haven't worked with you, but when I was involved in SMA, we did very much have a lot of common conversation with Cure SMA around how to develop our drugs and what would be good endpoints and get the input.
Yeah.
But I think to your question, how we can involve, or how patients can get involved, or patient groups can get involved, I do agree. I think, pharmaceutical companies in general, we're pretty good at reaching out and talking to patient groups. But I think, at least from my perspective, again, N of one, is I think it'll be important for patient groups to educate patients on what it takes for drug development. Because I think that patient groups don't really understand what it takes. Like, why does it take so long? Or what are you doing? Why are you dragging your feet? What is, and I think to really understand the core of drug development would be helpful. To understand the importance of placebo, to understand the importance of natural history and how it may or may not be able to replace the placebo arm. Like, those things, I think people don't, unless you're kind of entrenched in this, they don't really understand the importance of that. And I think that is something that I think patient groups can really help patients out there and make those connections. And I think that will really help this triangle, right, between patient groups, sponsors, and health authorities really kind of talk the same language.
And I would say one concrete thing is really, is there a disease registry that exists today for that specific disease? And if so, what can get done to enhance that? Or if it doesn't, that's the framework that NORD's put together and what c-path Institute to really say, okay, let's get that information, because that's really going to help move a field forward to really come up with therapies for that disease. And it's not only patients educating patients, but patients also educating others, pharmaceutical industry, et cetera, because the disease better than we do for some of these very rare diseases. You've been living with the disease, and that's really invaluable to move this forward. So there's already a framework in place to capture some of this, but is it really being done or not? And is it being done well? But that's the insights to help move this whole field forward.
And I think another area where patient groups could help is to the extent that there could be a hurdle for some sponsors to kind of contribute data to c-path or to this initiative, and to kind of share it broadly. I think patient groups articulating that that's kind of their expectation the expectation is that this data should be useful not just, for that specific trial and for a given sponsor, but it should be useful to the kind of, sort of the whole community, and should be submitted to larger repositories. I think that could help overcome some of those hurdles.
All right. Thank you, everyone, and thank you to our panelists. Appreciate it.
They've only begun trotting me.
Yeah. So we're going to break for lunch now. Food will be provided in the same area where breakfast was provided. If we've contacted you regarding a gluten-free meal, we ask that you check in with the registration desk. Thank you. Hi, Lindsay. Sorry, I have very cold hands. Thank you for joining our panel today.